See if this comes on. Yeah, so I'm going to be talking about regenerative medicine, building for the better of mankind. And that very much means I'm actually here to talk about Kåre. So Kåre is my father. He's a 70-year-old retired engineer. He was basically in sales his whole life. He was an early globalite, traveling the world, selling solutions for the telecom industry, basically. So I didn't really see that much of him early on because he was always in the US, basically. So very, very passionate about his professional life. And then later on, more passionate about family, I would say. So he's all about play. He's all about painting. It used to be a lot of landscapes, churches, birds, whatever. Right now, it's a lot of trolls. I think last month, it was a lot of cars. So he gets kind of focused in on painting different things. He's also a lot about exercise. He grew up an orienteering person, then lots of biking, hiking, now golfing. I think he's starting boxing for some odd reason. But also in terms of the play, like I have a daughter, she's 2.5. When we go there, she want to run to her grandfather directly. My brother and my sister, their kids, they always go and play with him. Or at least that used to be the case, because 2 years ago, diagnosed with Parkinson. So I'm sure you all know a little bit about Parkinson. It basically means that part of your brain has died. It's the part of your brain that produces dopamine, which is basically the oil and lubricant of the brain. So you need it very much for moving. It's very, very difficult to move around if you don't have it. You get very, very rigid postures. It's basically impossible to go to the loo. You can't even unzip or sip without medication. So it's quite a serious illness, especially when it comes to movement. But dopamine is also the thing that you actually affect when you take drugs, if you take drugs, or alcohol and stuff. So it's also part of many non-motoric functions. So if you get Parkinson, you get a lot of anxiety, you get stressed very easily, you get reduced lust. So my father, again, who's been traveling the world, he can't really even travel from Bålsta, which is 50 kilometers from here, to Stockholm without having to go peeing 10 times. He's really, really stressed. He can't really sleep the night before. And he can only really go here if he goes with mother. Like driving by himself to Stockholm, I think, hasn't really happened yet in the past 2 years. So it's quite a serious, debilitating disease. However, the good thing is that the hype around stem cells is really much coming to fruition now into real hope. Because the only way to cure a person like my father or Parkinson is to replace those dead cells in the brain, basically. So what you can do is you can use stem cells. And when I talk about stem cells today, I'm going to talk about pluripotent stem cells coming from the embryo. So basically what we all came from is the embryo. There are cells there that have two specific things that they can do. One thing is that they keep dividing. So you basically have an eternal source of cells with stem cells. The second thing is that they can become anything in the world. So you can define them to become brain cells. So you, in essence, can make spare parts for humans by using stem cells, directing them into neurons. So theoretically, we can now solve the riddle of Parkinson by creating stem cell therapy as a cure for Parkinson. But there's always the but. The tools to grow stem cells, define them into dopaminergic neurons, have been too poor. Stem cell researchers are still using like medieval tools to grow these kind of cells. It's not good enough. You don't get the cells you want. And that is where Biolamina comes into the story. So Biolamina is the first in the world to be able to make the glue of the body. This is something called laminin. It's a cell adhesion molecule. Basically, it's the protein that all cells grow on in the human body. So if you want to grow cells outside of the body, because you want to make spare parts, you want to make hearts, you want to make, if not brains, but at least nerve cells outside of the body, you want them to grow on their own natural substrate, because you trick them into feeling like they're at home, and if they're at home, they do what they're supposed to do. So it's very easy to grow them, and they actually become what they're supposed to become. So you get very, very functional cells, because up until now, when you're taking cells, there's a lot of variant. So if you're going to make these dopamine neurons, some cells are kind of dopamine neurons, some cells are definitely not dopamine neurons. So there's a large heterogeneity or variability in the cells that you make from stem cells, basically. So by using the Biolamina tools, you can just get like more homogeneous cells that really do what they're supposed to do, which is key for successful stem cell transplants. So this is the way a Parkinson patient looks like, completely without medication. My father is very much the same. He has this bent over position. He walks very, very slowly, almost impossible to turn around without medication, that is. So it's a very, very serious disease when it comes to these movements, especially. But there are some early trials. So the first stem cell therapy trial in Sweden was in 1987, and there are some patients that have remarkable results from this. So this is the same woman 5 years after transplantation here. This is no other medication but by having dopamine neurons transplanted to her brain. And I mean, she walks like any one of us. So it's really remarkable results using stem cell therapy for Parkinsonian patients. The problem is that this patient, it worked extremely well for. But there's a large variation when it comes to these patients that have been treated with the cells. So some patients, enormous effect. Other patients, no effect at all. Or actually, they come out worse than they were from the beginning. And one of the key reasons has to do with too poor cells being transplanted. They either don't survive or they don't produce dopamine, so they don't help the disease. So many of the cells that have been transplanted actually comes from aborted fetuses. So that comes with many problems. One is ethical. So you need 5 to 10 donated fetuses that have been donated within the past 48 hours just to treat one patient. So you can maybe treat one patient a week. And from a logistical perspective, it's dramatically difficult to do that, and also very, very costly. Ethically, very difficult ethical questions when it comes to taking 5 to 10 donated fetuses and take cells and transplant it into other patients. So this can never become a standard care. So the solution is really using these pluripotent stem cells that you can have as many cells as you want and define them into these neurons. And yes, this can now actually be done. So we are very much moving away from hype to hope to realism. So this is a paper that came out last year. Cell stem cell, it's like the New York Times of science. It's like the key journal that you want to publish things in if you're a stem cell researcher. That in itself says that this is good shit, basically, when it comes to research. And what it says is that they can now make cells that survive in the brain and make dopamine and basically cure Parkinson, at least in mouse models. But this is now being brought forth in clinical trials in Sweden in 2 years from now. This is a quote from Malin Parmar, who's one of the top 5 Parkinson researchers in the world. She happens to be in Sweden because Lund and Sweden has a very, very strong tradition when it comes to Parkinson research. So they think, and I'm a sales guy, these are not salespeople, they think that this can really become the first-line treatment for Parkinson. So very, very interesting times. Will my father benefit from this? Potentially not. But for other Parkinson patients, very, very interesting times and hopeful times. But not only that, so if we move away a little bit from stem cell therapy, the Nobel Prize in Physiology was given 2012 to Shinya Yamanaka, a Japanese researcher who have invented something called induced pluripotent stem cells, which basically means you can take a skin sample, turn it into stem cells out of anyone in here, basically. And if you can take any skin sample here and turn it into stem cells, it means we can make stem cells from all of you, turn them into neurons, and basically create personalized treatments for everyone. So coming back to my dad, that basically means that we can take a skin sample from him and we can make his own neurons. His own neurons. It's impossible to do otherwise. How on earth are you going to get brain cells out from my dad? Drilling a hole, taking part of his brain? I don't think so. So this is really a revolution where you can get neurons from the patient himself. It takes 3 weeks basically to get my dad's own neurons. And that means we can directly start to study the mechanisms and the characteristics of Parkinson's directly in the dish, in the lab, basically. But it also means that we can look at my dad's version of Parkinson's, because there's a large heterogeneity or variability among Parkinsonian patients. So my dad's version of Parkinson is different from many other types of Parkinson. And that's the only way to come up with other treatments, is to understand the key features of the disease for specific people. And that has an enormous effect when it comes to medication. So I haven't talked about the medication. So yes, there are good medications out there. L-Dopa, which was partly invented in Sweden, is something that basically becomes dopamine in your brain. And it works extremely well for most patients for some time. You have maybe 1 to 10 good years with that, but then you have 1 to 20 bad or horrible years, because this doesn't work long term. So there's a large need to have better medications, and you also have other medications, and you have to combine different concentrations and different combinations of different drugs. But what you can do now, when you have my dad's own brain cells in a dish here, you can start testing directly which is the optimal concentration for his type of brain cells. So you can basically make designer drugs for each individual Parkinsonian patient, which will have a large dramatic impact for this patient population. And it's as if that was not enough. So if we can make anyone's stem cells here, that means we can make my own stem cells. So I am the son of Kåre. I'm 38 years old. So we can make my own neurons. And when we have my own neurons, we can basically trick them into becoming old. So we could speed up time of my own brain cells, look at them in the dish and start comparing. What do they look like? Do they look like my dad's neurons? And if they do, does that mean that I will also get Parkinson? But even if it does, it means I can take very proactive precautions already now. You know, you can start exercising, which protects your brain. You can go on a specific diet, which protects your brain. But you can also choose what you're gonna do professionally. So I think that's my take home. You should follow your passion. Personally, I'm being part of coming up with the solutions for Parkinson, which I may one day get myself. So that's it. So building for the better of mankind and using stem cells have gone from hype to hope to realism. Thank you.
Thank you, Jesper. Do we have any questions in the audience for Jesper? Yes. Thank you.
Thank you. I was just thinking how much really inheritable this disease. For instance, you said once you can check your stem cells, and then how is really current technology, how is really practical in this method?
Yeah, in terms of just checking the neurons, well, that's easily done. So we could do it in our own lab at Biolamina. It would take us maybe a few weeks. So it's easy to check, and then you can check, how they look, morphology. You can check different things. So this is not something I made up that you can do. This is something that can be done, is done already. It's not really done for patients and father-son relationships, but it can be done, and it can be done now.
I have a question. You've been part of Biolamina from very early on. Mm-hmm. What are some of your
major... What have I done?
No, I know what you have done, Jesper. Amazing stuff. I'm curious about what are your major insights in moving with a company from a very startup phase into being more of an established company?
Right. Well, I think we're led by a father and son, Tryggvason, and the son is the CEO. I think he has one clear thing that he has told us all, like fail as much as possible, but just don't fail twice, basically. So I think we're testing a lot, so we're brave. I think if I compare to some other life science, high-tech startups within the biotech sector, I think they're much more cautious. Many of these companies are started by scientists. Scientists are cautious. We're also scientists, but I think we're a little bit braver. So I think one of the things we've done right is just testing lots of different things. I mean, it's kind of what every high-tech company does, but not within the life science industry. Even if it comes to marketing, sales, or even the product development or science, we keep testing continuously and keep failing, but we also do quite well quite quickly after the failure. So I think that's one thing. We're trying to take over the world. It's a mega small niche, but we have a very good potential for doing so. And I think we've come a little bit over the past 5 years in actually doing so. So that's part of what we've done.
Yes, we have more questions.
What are the side effects or drawbacks of stem cells transplantations?
So what are the drawbacks of stem cells?
Transplantations.
Well, so up until now, for instance, one of the problems have been that, if you don't control them well, there is a risk that they could become tumors in the brain. So there's a lot of work that goes into ensuring the safety. There's a lot of work that goes into ensuring that the cells actually do what they're supposed to do. So many cells have been transplanted that aren't the real cells that you think they are. Not because people knew they were the wrong cells, but you didn't have the right tools to measure the right biomarkers or the appearances that this will actually become through dopaminergic neurons or whatever other type, if it's heart cells or whatever you're transplanting. So having the right cells have also been very difficult. Maybe that's part of it.
Thank you very much for a very interesting seminar. And my question is, so how about the transplantation? Is it very difficult? Is it risky? And will that be a threshold for your success?
Yeah. So I'll be honest, I'm an engineer by training. I did a PhD in neurophysiology, but I'm not a clinician, so I can't speak that I know that by heart. But I met one of the lead clinicians 2 days ago in Cardiff. So it seems to be quite a straightforward transplant. Right now, it's very difficult when you take these fetal tissues, 5 to 10 donors. You need 3 to 6 donors per side of the brain, basically. So 5 to 10 donors in general. So that's a very tricky transplant. In general, you're going into the brain. But it has become much more of a routine. Now it's a stereotaxic treatment. So as far as I know, and again, coming back to the quote from these leading researchers, they think it's becoming, or may become, the first-line treatment, basically, meaning that they don't see it as that difficult if it works the way it should work. It's a fluffy answer, but I'm, again, engineer, not clinician.
Cool. Thanks a lot, Jesper.
Thank you.